Now, our next speaker is Peter Carberry from ICRISAT in India. Peter is talking about the environment, and in particular, he's going to talk about an important question, whether the environment is enough considered in the analysis of G by E by M.
Yeah. I'd like to start this talk off by saying how incredibly proud I am as the DDG of Research at ICRISAT, hosting you here along with our partners, ICAR and others, for this event. So really, well, congratulations to the team. I'm really proud of you. Hopefully after this presentation, you'll think I'm doing okay as well. Oops, let me go back. So, a couple of firsts. This is my first attendance at an interdrought conference. Because I thought you lot, this community of practice, were basically interested in breeding as a solution to drought or water deficit. So physiologists, plant physiologists, molecular biologists, breeders, and there wasn't really a forum for systems agronomists such as myself. And I've been really pleased with the talks I've seen this morning so far about and last night with Rob Bertram, this broader dimension and broader discussion around systems and how do we deal with drought. And the second first is that this is the first time I think in my career I've had breeding in a presentation title. I dealt with my career around water deficit and how farmers and managers might deal with water deficit, drought. And I guess for most of my career, I think crop improvement approach is a second order issue. It's not first order. I'm in interdrought here, so I could be in trouble after saying that, particularly after a great talk from Mark. So it's really pleasing to come to interdrought, to see the broader discussion and have the opportunity to talk about, well, why do I say that around first and second order issues around drought? I want to acknowledge Vincent Valdez, who's my co-author here. Vincent is the inspiration for this talk. When I first heard Vincent talk about some of the physiology phenotyping work he's doing at ICRISAT, I had some questions, and so that led to us having a conversation, and hopefully I'll be able to present some of the results of that conversation. So the talk is around about G by E by M, but saying maybe E is underestimated, and I acknowledge that both speakers before me actually talked about the E component, the soil water availability, etc. But most people, when they talk about environment, they think about climate, but it's also the soils and the biota, the pests, diseases that are inherent in any system, and also the systems that those crops are grown in. Now, I'm not going to talk about that systems. I'm going to stay away from my usual topic and stick to really the first three. So this is what I'm concerned with. I think you are. I mean, it's in the context of Australian farming, and this is the actual profit for a farmer in the Victorian Mallee in Australia over a 20-odd year period, 21-year period. And you get, even for a farm that's 5,000 hectares, it's a bit hard to talk about that when you're in India or you're working with your African colleagues. But in this 21-year period, that only 50% of years does this farmer make money, and 50% of years he lost a lot of money, or he lost a lot of money in a number of those years. So this is what we're trying to deal with. Even with the best technology that's available at the time, climate variability is critically important for dryland agriculture, and it makes dryland agriculture risky. It was really pleasing again to see Rob Bertram last night emphasise the whole dimension of risk. And what we've been able to do in Australia is an incredibly dry continent to grow crops, is that we've concentrated on trying to bring our knowledge about how to manage for climate variability together. And so here's an example of this same farmer, John and Ferrier, in 2011 being able to produce close to a 6-ton wheat crop in a decile 2 year. So this year was a drought, but this farmer produced a great crop because he brought to bear knowledge about the resource supply term, the soil and how much water was in the soil. He was able to protect that water through good agronomy in terms of managing his weeds. He used conservation tillage and good weed control. And you use knowledge about how the system works through decision support, through AppSim applied online, through yield profit, and some climate forecasting. And that gave him the confidence to invest in this crop in this year, put fertiliser on a crop, even though there was very low rainfall, particularly early in the season. So this is what we've got to be able to do, is try to give farmers and those that are helping farmers the knowledge systems to be able to manage for dry years. And a key thing about this is we've got to be able to communicate with this community, not the farming community in our community. This communication around risk. So this is some actual data from Kenya looking at climate smart agricultural practices. And it shows that even if you do the best job you can, use mulches, don't till, look after your weeds, etc., there's many years where even the best practice is going to fail and you're going to lose money. And we've got to be able to communicate this. We should not be standing in forums and saying that there's a solution to an inherently irreducible risk problem of climate variability. So I like framing agriculture and the practices that can drive farm performance in the framing of return versus risk, or output versus risk. And so many of the analysis we've done in the past is looking not just at the return, what the yield is, but what the riskiness of a certain practice might be. And I'm pleased when I wrote this paper in 2011, trying to volunteer what are the practices or technologies that might either move farmers up to the efficiency frontier, the risk efficiency frontier, might reduce risk or create new efficiency frontiers. But I did put harnessing G by E by M in there as an essential part of trying to deal with this big issue. And since I've joined ICRISAT, I've really come to appreciate the role that our crop improvement programs and all those that support it have done with our partners in India and in Africa in being able to deliver germplasm varieties and hybrids that do perform, that are increasing yield. And here's an example of chickpea in southern India. And that's supported by the contribution of our genomics, genetics, physiology, entomologists, etc., and being able to provide new tools that help support those crop improvement programs. So I want to concentrate now on this harnessing G by E by M. And many of you would have visited Vincent's LeasyScan facility, where he's able to quantify vapor transpiration and transpiration efficiency. And through that facility, he's been able to take different lines or germplasm and been able to quantify their water use. And can identify that there are particular lines that have water savings because of their physiological responses. So Vincent and team have published these new insights into transpiration efficiency, and I've heard some of the other speakers this morning also refer to this, Mark himself, and hypothesised that genetic gains via breeding can be made by manipulating these small amounts of water, either pre-anthesis or post-anthesis. So through the LeasyScan facility, it's showing that that water that can be saved, not used in the vegetative phase, could be used in grain filling, and that can lead to a productivity increase. So less water extraction in vegetative phase, more for the grain filling. And I want to acknowledge colleagues of mine from Australia, Graham Hammer and his team, for really pioneering this idea of gene to phenotype modelling, what Mark was talking about, in being able to put these ideas about what potential traits might do in terms of real-world systems. And so they've taken models like that, as Mark explained, and here's an example of slightly smaller sorghum leaves for the systems in Australia. And the graphs down the bottom here basically show the yield advantage of having those small leaves in that system. That work has been repeated by Yana Kolova and Vincent and the teams here for the rabi season in India, showing that if you have slightly smaller leaves, that many times under many environments that the yield there's a yield advantage for having that smaller leaf area, but it does come as a cost of the stover yield, which could be important for livestock. So it's good to see that trade-off or systems thinking coming in. So what I wanted to do when I first saw this with Vincent, I'm saying, but are you assured that saved water that's saved before enthesis in the vegetative phase is available to that same plant, that same crop in the reproductive phase? So I decided to pull up the sleeves, dust off APSIM and do some simulations. So this is looking at pearl millet in Sadore in Niger over a time course of years, 1983 to 96, and looking at what the yield might be and whether or not, well, actually this is me having a go at trying to run APSIM again after about 7 or 8 years. So it was a struggle to get there, but I finally got there. So very small changes in leaf area index or leaf area development. So here's the difference at leaf area index at flowering, what that might mean in terms of benefit during grain fill. And the model confirmed what the hypothesis was, that if you don't use the water pre-enthesis, and that water is available during grain fill, and you'll, in most of the years, in 11 out of those 14 years, you'll get a yield benefit. As clearly 1995, there was not a benefit, and I've gone to have a look at what's happening there, and it happened to be around tiller death, tiller dynamics that caused a different aspect in that year. It was quite a dry year. Okay, so it works, but does it? Millet in Niger in West Africa is generally grown with an intercrop, cowpea. But what happens if you put cowpea in the system that also has access to that water? And you can see that there's still a benefit in a number of years, but it's dropped from 11 out of 14 years to only 8 out of 14 years that you'll get a benefit because the cowpea has access to that saved water. What about in a real world system, you have weeds. So weeds germinate, and it takes some time for a farmer to be able to manage those weeds. So what happens if the weeds germinate, and it takes 3 weeks for that farmer to actually weed that crop and only weed it once? And you can see again, it's in 8 out of 14 years, you get a yield benefit from that saved water because the weeds will also use some of that water. What happens if the weeds germinate and you don't get around to weeding because you can't weed all your fields? Then the benefit's only 6 out of 14 years because the reality is there's other parts of the system that will use that water. And lastly, all those simulations were done with 60 units of nitrogen put on. Farmers in West Africa don't apply fertilizer at that level. What happens if you run it with no fertilizer? Well, you see no benefit. Well, sorry, I'm overstating that. In 2 out of 14 years, you get a benefit from smaller leaves. The point I'm trying to make here is that fine if you're in a highly managed system where you can control the other dimensions of the system, then saved water can be used post-anthesis. But many of the ecologies that ICRISAT works in are much more complex than that. Weeds, intercrops are part of the system. So you can't assume that that approach, that particular trait, will be manifest in these systems. And there's a range of these environmental, what I'm calling part of the E term, that you have to consider if you're going to look at particular traits in these smallholder systems. So I've talked about weeds, the extreme temperatures that the millets and sorghum that ICRISAT's responsible are grown in very harsh environments. And you see even millet in this slide here has failed seed set because of the high air temperatures. A big issue I see when anybody that's travelled through West Africa or Rajasthan or Southern Africa in these more marginal environments is crop establishment, as you don't see perfect stands. And clearly soil fertility is an issue as well. The crops aren't grown to their potential. And to me, the E term is so dependent on the soil profile. And I'm really pleased to see the other speakers also talking about soils and characterising soils for water holding capacity, hopefully for fertility, for subsoil constraints is also part of the E term. Find this. Where am I pointing? So I'm moving on to soil fertility. So this is some simulation work done on climate change by John Dimes. The top two curves here represent what the yield distribution might be for maize grown in Bulawayo in Zimbabwe under current conditions, but high fertility. And then with climate change impacting, you'll have a reduced yield because of that impact. And this is what most simulations of our climate change impacts do. They take potential yield and look at the impact. But look at the results when John simulates this same scenario under current conditions that most smallholder farmers grow maize in Zimbabwe. Like I say, there's, except at the very low end, low yields, there's no difference in terms of what happens in 2050 relative to today in terms of the probability of achieving a certain yield. So the key thing is, what is the impact of climate change if we can't have an impact on how crops are grown today? And John has been quite controversial in this paper that he's written about saying adaptation by breeding for drought is misplaced probably in these ecologies because of the big impact of soil fertility. And this is backed up somewhat by some great work that was done by Steve Twomlow and others by ICRISAT in Southern Africa and Zimbabwe during a drought, the 2003-2004 season, where there was a relief program that distributed seed of hybrids and fertilizer to 160,000 farmers or farm families. And Steve went in behind that and implemented trials of where they put just the hybrid with no fertilizer, or the fertilizer was applied to the OPVs, or they put both. And you can see that the benefit from this survey of farmer practice and using technologies such as hybrids or fertilizer, that the hybrid seed did give a benefit over the OPV, but not as much as applying microdosing, 17 units of nitrogen to the OPV. And of course, if you put the hybrid and the fertilizer on together, you get a greater benefit. So the role of importance of looking at soil fertility relative to genetics into this system was well played out in this survey. So I put this up. It's really interesting to see the number of Australians at this venue and even in the talk so far. And one of the reasons for that is the Australian ecology is very similar to the target ecology of the semi-arid tropics for ICRISAT. This map shows that that northern Australia is very similar to West Africa, the Sahel savanna of West Africa, and also to southern Africa. And I've got arrows there from the points where I've spent much of my career working. The first 5 years in Katherine in the Northern Territory, and subsequent to that onto the Darling Downs. And why have I put that up is that when I first started working in that semi-arid tropical environment, we saw the biggest constraint to crop production was crop establishment. But farmers struggled to get crops out of the ground, let alone once they're established, what the impact of water deficit is. So the first graph here shows the probability of sowing opportunities in this environment. You can see that in 50% of years, you only get something in the order of 15 days to be able to plant a crop. A number of those days are going to be impacted by high temperature and others by crusting. We did work to look at the impact of high soil surface temperatures on maize mortality or seedling growth. And you can see once the soil surface gets above 50 degrees, plants are going to die as they come out of the ground. Then if you put that into a model, you can see that the plant population in this environment is going to be reduced by high soil surface temperatures as the crops emerge. And this last graph here shows that once they get out of the ground, then if they're subject to early season water deficit, then they can also die. So this is a representation of a cumulative water deficit over time and plant survival. 1 means there's no death, 0 means that all the plants die, and this is the difference between maize, sorghum, and kanaf. So this is data showing that it's not just a matter of planting or sowing a crop in these environments, it's getting the crops established, getting to the point where they can reach grain filling. And so those of you that work in West Africa or in Rajasthan, you will see situations like this on farms, where it's not the plant that's well established that's running into water deficit during grain fill. It's the fact that there's no plants there because crop establishment didn't occur. This is a major constraint in these environments, and it's part of the E term that we want to see. And I think it's mostly ignored in systems analyses. In fact, all those equations I put up there are in the AppSim model, but they're not turned on when you download it. So I don't think that the many of the analyses done, even though this capability is there, incorporate issues around crop establishment and early season water deficit. And I do worry about the quality of simulations that are done for these environments without properly taking this into account. I'm a bit cheeky here by putting up a photo of Rob's talk last night, and when he talked about the impact of the El Nino and adaptation to it needs to deal with climate variability. I was taken by this picture because most people would look at the plants and say, well, they're stressed. I look at that picture and look at the fact that there's no plants there because the crops didn't establish or the plants didn't establish. That's a key issue when you're talking about drought. Okay, that same simulation, one of the key issues is that many of these systems are not water constrained. Here I am in an interdrought conference and I'm telling you that the systems of many of their target ecologies aren't water constrained. This simulation showed that there's significant amounts of water left in the profile at the end of the season because of fertility is not there, or the plant population is not there, or the vigor is not there to be able to extract the water that's available to the plant. And this is not new. You can look at the work of Charles Builders, Bill Payne, Ramajita Tabo, Steve Twomlow, John Domes, is showing that this has been around for a while, that basically under the current systems that farmers use in these ecologies, the water resource is not used. Bill's saying that you can easily increase the population and the fertility without having a risk of running out of water in most of these ecologies. And this leads us to the work that's back to the work we did in Australia, some work that Anthony Whitbread did in the Mallee, showing if black's bad, white's good, that if you have low soil water at sowing and you have a late planting opportunity, then your prospects for a good crop are low. But if you have good soil water at sowing and an early planting opportunity, then you're pretty well assured of a crop. This is a much better predictor of how the season's going to play out as opposed to a weather or climate forecast. And so it's important, if you're going to do that, to understand what the water holding capacity of your soils are. You can use simple techniques that farmers in Australia are using to characterising the upper limit and the crop lower limit of their soils. These simple tools are being used by farmers and agronomists to service and provide information to farmers. And you can generate information not for a typical soil in a region, but for an actual field or a paddock on a farm. So many farmers have gone and characterised many of their paddocks, so they understand the difference in terms of the water holding capacity on their farm. And the industry is spending a lot of time training people around how important it is to understand soil water as a way of managing climate variability. And so we're lucky enough now in Australia because those data that's been collected on farms, generated by industry, by farmers, by the researchers, is being put into a public database. You can download soil characterisation for all the cropping belt of Australia based on that information. I put this up before I saw the talks this morning, but how many breeding or G by E by M trials measure plant available water content or available water? And it was really good to see that that actually is happening. My last slide is trying to deal with the biotic pressure. And this is a little story from Australia and probably replicated elsewhere where BT cottons come in. is that we have a very well-known modeler that built a model for cotton in Australia, Brian Hearn, and Mike Bench followed. And what they realised when BT cotton came in is their understanding of the physiology and the behaviour of cotton had to change. Their assumption was those early fruit didn't set bolls and produce contribute to productive yield. When BT came in, they realised that those bolls could play an incredible role in building yield, and it was just that there was a low-level pest pressure there that they didn't know about because they weren't spraying early enough to capture that. So the physiology of the plant had to change in terms of the models and understanding of how the plant grew. And I just wonder how much of that is evident in our farming systems, that there's a low-level pest pressure that might be having an impact in suppressing yield. So, to conclude, I think, as I said at the start, I wasn't sure that breeding for drought is a first-order issue. Of course, breeding can contribute to better performance under drought, and I see that in our research done at ICRISAT and our crop improvement program. We need to understand the G by E by M interactions, and it's critical. And E has to be broader than just rainfall. And also, a point here is that it has to be broader than just crop and a crop model. Once you go out into systems where there's intercrops, where there's weeds, where there's other factors come into play, a crop model doesn't suffice. You have to have a cropping systems model, or once you get into systems with livestock, you have to have a farming systems model to be able to look at these dimensions. I think there's likely too much emphasis on the crop demand term relative to the resource supply term in trying to deal with drought. E is more than rainfall, as I've tried to point out, and a systems context is essential. With that, I'll say thank you.
Many thanks, Peter, for the interesting and practical talk.